Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with the Standard 12 Biology Chapter 11, that is Biotechnology, Principles and Processes. So in the last two videos, we have learnt about the restriction enzymes or the restriction endonucleases, which are the tools for DNA, recombinant DNA technology. So in the last video, we have seen these exonucleases and endonucleases. Then we have learned what are palindromes or palindromic sequence. Then now in today's video, we are going to learn about the sticky ends and the blunt ends. So let us begin. Biotechnology, principles and processes, blunt end and sticky ends. So what are these blunt end and sticky ends? Now when restriction endonucleases or when an restriction enzyme cuts a fragment of DNA, it can either form a blunt end or a sticky end. Now suppose this is a DNA fragment and a restriction enzyme has cut it. Now this will form a blunt end like this. It will form a blunt end. Whereas there are some other type of restriction enzymes which cuts the DNA in a staggered manner. Okay, so they will form something like this. The DNA fragments will be cut like this, and these will form these portions which are called as the sticky ends. Okay, so let us begin. Now many restriction enzymes make a simple double stranded cut in the middle of the recognition sequence, resulting in a blunt end. So as we can see, this is the blunt end. Then in other restriction enzymes, the two DNA strands are not cut exactly at the same position. So one strand is cut over here, whereas the other strand is cut over here. Okay, so as we can see, one strand is cut over here, the other one is cut over some distance. Then instead the cut is staggered. So instead the cut is staggered, usually by 2 or 4 nucleotides. So if one cut is taking place over here, the next cut will be 2 to 4 nucleotides apart. So that the resulting DNA fragments have a short single stranded overhangs at each end. So as we can see, there are some these, the short stranded overhangs, single stranded overhangs at both the ends. Now these are called as the sticky ends, as base pairing between them can stick the DNA molecules back together. So these sticky ends can again base pair with the DNA molecules of other. Then the stickiness of ends facilitate the action of enzyme DNA ligase. So let us see over here. Now these are the steps in formation of recombinant DNA by the action of restriction endonuclease enzyme, that is E. coli enzyme. So here we have two DNA strands. This one is the vector DNA and this is a foreign DNA. So here the restriction enzyme E. coli is used to cut these fragments. So what is happening over here? The restriction enzyme is cutting the strand over here. So from here it will be cut and over here. So and this strand is cut from here to here. So now what is happening is the DNA will have this portion, single stranded portion, and this single stranded portion, okay, as we can see here, which will form the sticky ends of the DNA and which can be then ligated. The DNA fragments can be joined at the sticky end. So as we can see, here we have TTAA sequence and here we have AATT, which are the complementary nitrogenous bases, and they can be joined together to form the recombinant DNA. So this is how the restriction enzymes cut a DNA fragment and can form either blunt end or a sticky end. Then talking about the nomenclature of the restriction enzymes, that is how we can name these different restriction enzymes. So the rules for nomenclature of restriction enzyme are: the first letter of the name comes from the genus name. So we learn this by looking at the examples. Now the first one which we have is E. coli. Enzyme name is E. coli. Now how? Now first letter of the name comes from the genus name. So what is the genus name of source of organism? It is Escherichia. Okay, it is Escherichia. So first E will take as the first letter. So E. Then the second two letters comes from the species of the prokaryotic cell from which they were isolated. So the species is the coli. So from here the second two letters, so C and O will be taken. So E for Escherichia, C and O from the species, E. co. Then after following these three letters, the strain is sometimes represented. So the next thing is the strain. Now this strain was RY13. So from here we have taken the R. So Eco R. And then following the strain, the Roman numbers following the name indicates the order in which the enzyme were isolated from that strain of bacteria. So the order of isolation from the bacterial strain was 1. So E. coli 1 or E. coli. Then talking about the next one, which is Hind 2. Okay, so Hind 2, the genus name of the source organism is Haemophilus. So H we have taken from here. Then the species name was Influenzae. So I and N, H I N. Then the strain is D. So Hind, HIND, and the order was 2. So Hind 2. Okay, so this is how we name the different enzymes. Then also talking about this sticky end and blunt end, how they are formed. So let us see the production of blunt end. This is a DNA fragment and this is the restriction enzyme used, which will simply cut the DNA in the middle and form two blunt ends. Then production of sticky ends. This is the DNA strand and here we have taken the E. coli restriction enzyme, and here as we can see this will form the sticky ends as it will cut the DNA fragments at two different positions. So this is all. Thank you for watching my video. Please like, share and subscribe to the Bodhi Academy.